Good evening, and welcome to the first Longwood Seminar of 2012. We're delighted to have so many people with us tonight. I'm Gina Vild. I'm the Associate Dean for Communications and External Relations at Harvard Medical School. There are so many familiar faces. If you've attended a Longwood seminar in the past, would you raise your hand? Fabulous. We're thrilled to have you back. And if this is your first Longwood seminar, we hope you will attend our other 3 programs and continue to come back in the years to come. I hope all of you are friends of ours on Facebook. You may know that our program this year was chosen by our Facebook friends. We had 500 votes in total, and tonight's event was one of the most popular. If you're not a friend of ours on Facebook, we invite you to join. You not only will receive information about the Longwood seminars in the future and more this year, but we will soon be expanding our page to include information on a variety of other Harvard Medical School programs. We have 3 other fascinating programs this year, in addition to tonight's program on Alzheimer's disease. The Science of Emotion will be held on March 20th. Cancer Genetics will be on April 3rd. And Stem Cells will be on April 24th. Now, just a few announcements before we begin the program. We will be videotaping the program, so you can share that information with your friends and relatives who weren't able to attend tonight. And we will be creating a podcast out of this evening's program for the first time to give you some options. If you are in one of the first rows and would rather not appear on our video, I would suggest you move to the back or to the balcony. Also, we are going green. So the Longwood Seminars is part of our Going Green effort to support Harvard's sustainability goals. We're going to be environmentally friendly, and therefore we printed limited copies of our handout material, and I understand it's all gone, but you can obtain this also online. Our survey also this year will be part of our green initiative, and rather than present you with a survey, for those of you who are familiar with it, at the end of each program, we'll be giving it to you at the end of the 4th program electronically. And for those who attend 3 or more Longwood seminars, just a reminder that you can receive a certificate of completion. We actually had a call today from someone who asked what they're able to practice after they receive our certificate. So I don't think it's going to open many doors at medical school, but it might be valuable for something, or at least it will look good on your wall. Also, professional development points can be earned by teachers if you attend 4 or more seminars. These are available at the check-in table in the lobby. We'll be distributing index cards. I know you'll have many questions, and you'll be able to pass the cards to our team of volunteers who are working tonight. And finally, please turn your cell phones off out of courtesy to our speakers. Tonight's seminar is called Patience and Patience, Understanding the Spectrum of Alzheimer's Disease. As our life expectancy lengthens, we are increasingly touched by the incidence of Alzheimer's disease in one way or another. And there are probably few of us in the audience tonight who haven't in some way been touched by it, through a family, a friend, or an acquaintance. Someone who's received this life-changing diagnosis. Fortunately for all of us, researchers at Harvard Medical School and beyond are continually learning more about what causes this debilitating condition and discovering new ways to help patients and their families cope with the symptoms. We're fortunate to have many experts here at Harvard Medical School whose research is advancing Alzheimer's disease, and tonight you'll hear from a few of them. Dr. Risa Sperling is an associate professor of neurology at Harvard Medical School. She is also the director of the Center for Alzheimer's Research and Treatment at Brigham and Women's Hospital. Dr. Ruth Kandel is an assistant professor of medicine at Harvard Medical School, associate professor of medicine at Beth Israel Deaconess Medical Center, and director of the Memory Disorders Clinic at Hebrew Senior Life. But first you'll hear from Dr. Adrian Ivinson. Dr. Ivinson is the director of the Harvard Neurodiscovery Center here at Harvard Medical School. To add to this evening's educational experience, Dr. Ivinson has brought with him a human brain. Anyone who's interested in viewing it is welcome to meet with him after the program at the front of the room. The brain tissue will remain covered and offer you a unique experience. So again, thank you for joining us, and Dr. Ivinson.
Thanks so much. Thank you very much. What a great turnout. Thanks for joining us this evening. I would just like to correct one thing Gina said. I actually brought two brains, but one of them stays where it belongs and the other one's in the box. So we'll only be looking at the one of them. Okay, let's see, where are we? Number one. Oh, we're off. So I guess my job this evening is to really set the scene for our two subsequent speakers who are going to really give you the substance of this evening. They're both neurologists, they both do research, they both see patients. And as one of my daughters would say to me, he is a doctor, but he's not a proper doctor. So I'm not the proper doctor, they're the proper doctors. And so what I'm going to do is I'm going to race through some sort of basic background on the brain, a little bit about Alzheimer's, and then a little bit about some of the approaches we use for research, and then I'll turn it over to the experts. So very briefly, that's what I've put up here, what I will cover, and then Dr. Sperling will look at early diagnosis, and she's one of the world's imaging experts, so you'll hear a lot about that, I'm sure. And then Ruth will talk to us about disease progression, different stages of Alzheimer's disease, and really the sort of sharp end of caring for people and coping with Alzheimer's disease. Okay, so let's get started. This is your brain. Okay, it's just a phenomenal organ. So I think quite often we think of the brain as this sort of fairly quiet, dusty thing in our head, that does the occasional bit of thinking for us. You should actually think of it as a sort of athlete's muscle, working at full tilt. It's only about 2 or 3% of your body weight, but it uses 20% of your blood supply. So it's a hard working, fast, active, dynamic, kinetic organ. It's got about all the organs in our body, it's made up of cells. And in the case of the brain, they're very specialized cells called neurons. It's got about 100 billion, so that's billion with a B, neurons in there. And it's just phenomenal to think about how this wonderful organ comes about. How does it develop? How does it do what it does? Those 100 billion neurons, on average, make connections about 1,000 times to 1,000 other neurons or to muscles or to other things they control. So that means 100 trillion connections. So think of it as the most complicated, complex, wonderful network. And it's the network that really makes the brain do what it can do. It's the way it connects up. Okay, so what's at the... if you excuse the bad analogy, what's at the heart of the brain? So here's my very bad drawing of a neuron. I'm pretty sure they don't look like this, but this is my sketch of a neuron. So it's these cells, they have a cell body. But the thing that's special about neurons is these funny appendages they have. And you can see I've marked two types. There are these dendrites. They bring messages into the cell. Then it's got this long one called an axon. And it's the axon that generally takes a message out. And you can see it sort of divides at the end, the axon terminal, and it passes that message or that signal on to whatever it's connected to all those other cells. So this is the very basic component. This is actually what they look like under the microscope. So you can see actually the complexity of it is much more than my bad diagram. Each of these little knobs and bumps here is probably a point of connection. And those points of connection can come and go depending on how you use it, how you use your brain, how you use the cells. So let's go back to my neuron. So this is what we're talking about. Input from other cells coming down these dendrites. And then the message goes along the axon. And then it connects out and it sends the message elsewhere. And I'd just like to point out it took me about 3 days to do that animation. I should have better things to do. Okay, this is someone else, an artist's impression of what you're looking at. So think of the brain as this very dense network of cells shooting messages along its axons, connecting to other cells, passing that message on. But of course, think of it on a massive scale. 100 billion of these cells making 1,000 connections each. And think of the complexity with which it has to do that. You don't want a message leaving one cell and just randomly connecting to other cells. It has to be very specific and precise. So you can see the complexity here. And if I freeze this at, let's say, here, you see right up here, this is, again, an artist's impression, but you see this point here? So here's an axon coming down from another cell, and here it is just almost touching, barely kissing this other cell. So the message will come down that axon as an electrical message, it'll reach the end, and almost always it'll be transmitted across that little gap as a chemical message. And that's how the signal will be passed on to the next cell. There we go. And here's a close-up of the same animation. So this is the synapse. This is how the cells talk to each other. And ultimately, when things go wrong with our brain, it's because things are not working well at the synapse. Messages are not being transferred appropriately from one cell to another. Now, it could be that it's firing too fast, and you've got overactivity. But in the case of Alzheimer's, what's happening is this signal is breaking down. These cells are no longer able to talk to each other. And when that happens, ultimately, the cell that can't communicate will sort of recognize it can't communicate and will die, and you get cell loss. So as Alzheimer's goes along, it starts off as a process where, and we'll talk a little bit about this, things are going wrong that ultimately cause this connection to fail and ultimately cause these cells to die. And they die on a massive scale over the course of the disease. Okay, now I'm going to jump briefly in a different direction. So here's a very simple graph showing you the number of Americans over the age of 65, going all the way back to 1900. So back in 1900, there were about 1 or 2 Americans over the age of 65. And it's a fairly steady climb over the next century. So here we are today, and there are about 40-odd million Americans over the age of 65. The growth of this growth here is down to 2 things. It's because we're living longer. Back in 1900, average life expectancy at birth would be about 50 years old. Now it's about 80. So we're living longer, so more of us are getting into this 65-plus category. But it's also because the population's growing. And look where we are now. So this is the middle of 2000. Here's 2010, 2012, right here on this inflection point. And you'll notice that curve does something very dramatic. After a century of steady climb, it suddenly clips up. It's a much steeper gradient. That's the baby boomer generation coming in, hitting the age 65, and you can see how it's projected to rise. So why do I tell you this? Well, sadly, 10% of those over the age of 65 have Alzheimer's disease. So the 4 to 5 million Americans that are suffering with Alzheimer's disease now is really the tip of the iceberg. What we're going to see, if we don't do something about this, is a doubling or a tripling over the next 30 or 40 years. And that should really take your breath away. It's not just a US problem, it's a global problem. If we look around the world, this is probably terribly underreported, but it's estimated there are about 35 million people with Alzheimer's disease in the world. And this is the projected growth. So again, 2 to 3 times a tripling over the next 40 years. Where are they? Well, most of them are in low and middle-income countries, which again is something worrying because it's difficult enough dealing with Alzheimer's disease in a wealthy country like the States. But in countries that don't have that infrastructure, this is going to be it's like a sort of tidal wave waiting to happen. And here's one last number to leave you with, just in case you're in any doubt as to the challenge of Alzheimer's disease. In 2010, the global economic cost of this one disease was $600 billion, and that is a staggering 1% of the total global GDP, gross domestic product. So the total value put out by the entire planet is gobbled up, 1% of it is gobbled up by this disease. So my point here is we better do something about it. Okay, Alzheimer's disease. Risa and Ruth will go into this in more detail, I'm sure, but we all pretty much know what Alzheimer's disease is. It starts off as memory loss. It can be quite mild, but this disease progresses over a decade or thereabouts, and it ends up in a very significant deterioration. And again, the point here is a simple one. This is not aging. This is a specific disease. Maybe a generation ago, maybe 30, 40 years ago, we'd say, well, that's what happens when you get old. Not necessarily. This is a specific disease. We should be able to do something about it. Aging, now that's another problem. Okay, so what might this look like? I just thought, a couple of examples to sort of drive it home as to what Alzheimer's disease might feel like, or how might we recognize it. So if I sat one of you down at a table and gave you a pen and a bit of paper and said, draw me the face of a clock and put on that clock the time 4:45, hopefully you would do this. But actually, it's quite a sort of complicated task. You've got to know numbers. You've got to know how to put those numbers. You've got to know conceptually when the hand of the clock points at a 9, that doesn't mean to say 9 o'clock. That actually means 45 minutes. There's some complexity in that. When patients come in at various stages of Alzheimer's disease, this is one of the tests that neurologists might give them just to see how they're getting on, to see how they're coping, a quick test. And here's some results from a colleague of ours, Dr. Selko, who gave us this. And you can see that, this patient here, they were all given the same challenge, 4:45. This patient here has sort of got it vaguely right, 3:25, but, the clock looks pretty good. But look at these other ones. You can sort of almost feel the frustration that these patients are feeling where they just cannot get this right. It's a clock, they've seen it a million times, but they just can't quite get that out. I thought I'd give you just one other example of how Alzheimer's disease might affect someone, and it's a very sort of visual effect, I think, that'll help you understand it. This fellow here is a handsome young man in the 1960s, is William Utemohlen. He's an American, an artist who lived in London for most of his adult life, and he used to paint all sorts of things. He's a terrific artist, but occasionally he would stop and do a self-portrait, and this is one of his self-portraits. Sadly, Mr. Utemohlen came down with Alzheimer's disease in 1996, and he continued to do self-portraits whilst he dealt with this disease, and you can see the results here as we go through these roughly a year apart, until this was the last self-portrait he attempted. Sadly, he died a couple of years ago, and you can just look at these and you can see what this disease did to this person whose life was all about portraying images and perspective and color, and you can see how he struggled. We actually were fortunate enough to have a lot of his paintings brought over here for a show a few years ago, and his wife came over. She's actually an art critic, and she reflected that she wondered whether when he was painting this, he was painting himself almost behind bars as if he felt imprisoned. She commented that when he stopped painting, he would say to her, "I know there should be a nose, but I don't know where to put it." Now that's pretty devastating. Okay. So what are we going to do about this in terms of research? That's all the bad news. Let's try and be a bit more optimistic. Well, I've sort of generally broken this down into 3 areas of research. This is a bit of a simplification, and when we're doing this correctly, it doesn't actually look like 3 different disciplines doing different things. It's all connected, it's all feeding back. But it's not unreasonable to think of 3 areas of research. So let's just take a quick look at these. Basic research. So this is when we're asking very fundamental questions about the nervous system. So I said 100 billion neurons. Well, where did they come from? The development through the fetus and into early life and then early adult life, how do those 100 billion neurons come to be? How do they position themselves so precisely just in the right spot? How do they make connections to the other neurons that they need to make connections to? And how do they remove the connections that they shouldn't be making? It's a constant sort of process. These are the sorts of questions you might ask in the field of basic research. For example, we looked at this neuron. Why does it actually look like this? What causes one of these dendrites, one of these arms, to move in this direction? Why didn't this one head down this way? What were the triggers and the pulls and the signals and the messages and the chemicals that encouraged it to move in that direction? Because this is not a random thing. It sort of looks random to us when we put it up here, but it's not at all random. So how does it go about making those networks? Most complicated networks. And when we look back at the synapse, I said that the message jumps across this gap. Well, what exactly does that mean? How does it jump across? What are the chemicals? How many of those chemical molecules are there? What's the diversity of the chemicals? What happens if you change the chemicals slightly? Can the message go both ways? Is it always chemistry? Sometimes is it electrical? Are the different connections of different strengths? How does it assimilate a strong signal and a poor signal? So these are the sorts of questions that we're asking in terms of the very basic fundamental understandings of neuroscience. If we don't do that, then we don't have anything to build on for the next stages of research. If we can't try and describe the universe of the brain right from its earliest developmental stages through into aging, if we can't explain some of these fundamental basic questions, then how are we going to fix them when they go wrong? So if you like, the basic neuroscience, the fundamental neuroscience, I didn't mention Alzheimer's in any of these questions about basic neuroscience, but if we don't do this, then we're sort of building our clinical research on it's a house of sand. It's got no foundation. So basic research, if you like, is the engine that drives us. Translational research then tries to do something a little different. It says, how can I take this basic fundamental understanding of the nervous system, and how can I start applying it to specific questions? So in case of Alzheimer's disease, what are the risk factors? If 10% of people over 65 have Alzheimer's disease, what's special about them? Or what's special about the 90%? What's special about the person who gets the disease and it seems to move very rapidly compared to the person who seems to get the disease and it progresses very slowly? We know that some of these risk factors are in the environment, but we know that a lot of them are inherited or genetic. But it's only very rarely that the inherited component, the genetic component, is precisely or simply inherited, like, for example, cystic fibrosis or Duchenne muscular dystrophy, where you have one gene, the gene goes wrong, you get the disease. That's not what happens in Alzheimer's disease. There's probably 100 genes, each of which carries a small part of the risk. And you can think of it as an old-fashioned weighing scales. We've got risk genes on one side, we've got protective genes on the other side. If you've got a balance of more protective genes, maybe you're in the 90%. If you've got a balance of more risk-bearing genes, maybe you're in the 10%. So how do we understand that at a population level? How do we understand it at an individual level? We can't yet do that for any of us. We can't pick someone out here and test all those genes and tell them what their risk is. But we're beginning to make inroads into that. We're beginning to describe some of that genetic risk. A lot of what we do is all about developing better tools and technologies, and Dr. Sperling in particular is at the forefront of this and one of the most exciting tools that we use. So I won't steal her thunder. But building tools, whether it's microscopes, whether it's new chemical approaches, whether it's putting together the robotics that allow us to do a very high throughput, massive scale of research. Instead of pipetting, one tube, one test tube at a time, can we do it 100,000 tests at a time? Can we get the statistical expertise to help us really look at these massive scale data and decide what's the knowledge inside the data? And we spend a lot of time thinking about disease mechanism. So in Alzheimer's disease, I've described some of the things that go wrong, but I haven't really told you anything about mechanistically, fundamentally, what is it that causes that thing to go wrong. When I said that the cells die, why do they die? When I said that the synapse, the connection between cells breaks down, well, why? So we spend a lot of time thinking about the underlying mechanism. We know what the symptoms of Alzheimer's are, but what mechanistically is happening? So for example, when someone dies of Alzheimer's disease, if they're good enough to donate their brain to us, and many, many do, and we put a slice of that brain and we stain it and we put it under a microscope, we see these things which are very odd and you wouldn't see in an otherwise healthy brain. This is a plaque made up of little pieces of protein, a protein called amyloid. And it's a particular little slice of this protein, and it's millions of copies of it. And they tend to clump together, and they sort of gum up the works. It's a technical term. We also see these things called tangles. This is a different protein. This one's tau. This one is actually inside a cell. This one is outside the cell. But these things seem to closely correlate with Alzheimer's disease. So we spend a lot of time asking, did they cause the disease? Or did the disease cause these things to happen? Where did it happen in that cascade of events? We actually know quite a lot about this now. We know how these proteins come about. We know where they start. We know how they build up. And some very, very recent data that's only emerging last week and this week is telling us a bit about how they spread through the brain. So we're learning an awful lot about this, looking at the pathology of the disease and trying to figure out precisely why that happens. If we go back to our model, my beautiful neuron, this is one way of thinking of the plaque. This was another 3-day animation exercise. So here's our plaque building up around this cell. And we know that when a plaque builds up, one of these, this was the red-colored thing in that previous slide, we know that it has consequences here. We don't know exactly what the consequences are, but we know that it ends up in limiting the ability of this cell to communicate, and ultimately this cell will die. So we're trying to link the development of this plaque to the death of the cell. That would tell us something about mechanism. Let's just look at one other example of mechanism in my last minute. So here we're just looking at a graph that says, what if we had some scale that said a healthy brain and age? And what if a patient came to us who said, I think I'm getting the first signs or symptoms of Alzheimer's. I don't know, I'm not sure, but I seem to be forgetting more than I used to. It doesn't feel right. Okay. And then we would say, okay, so when first symptoms appear, let's just arbitrarily say that that means they've got a brain of this healthiness, whatever that means. Okay. We then follow this patient for maybe 10 years, and we see that when they come back and they do that exercise with the clock and a lot of other tests and exercises, we can see that their ability, their capacity, their cognitive capacity, their memory is declining. And it's declined this much. What we now know is that this is really just the tail end of the problem. What's actually been happening is this. What we now know is that 100% of that person's brain health, if I can put it that way, is up here. And it was 20 years or 10 or 30 years, we're not sure, prior to the first symptoms that actually the damage started occurring. But they didn't know about it. The brain compensates. It's got 100 billion cells, and it finds another way around the problem. So this cell dies, and this pathway is weaker. It finds a way around it. And we compensate ourselves. When I forget things, I write them down. So we compensate both in our heads and we compensate outside in the world. But actually, this is the disease process. And so now what we're doing is we're thinking, well, what was the triggering event here? Not so much here. There's too much damage done. What was the triggering event here, and can we prevent it? And then finally, clinical research. So, and Ruth will be talking about this. So really, what does disease progression look like? How can we really understand how this behaves in an individual and also in the Alzheimer's population? How do we cope with the disease? How do we test new drugs? That's what we want to prevent the disease. So in terms of clinical research, there are all these clinical trials going on. At the moment, I looked it up yesterday, it said there were 257 ongoing clinical trials for Alzheimer's disease. There's a whole host of things in there. They're testing devices, dietary supplements, exercise regimens, everything. But they're also looking for new drugs. And in fact, there are a couple of very important clinical trials that are due to finish this year, and we all have our fingers crossed and knocking on wood that they show some promise. These clinical trials take an awful long time to do. Usually, phase 1, phase 2, phase 3 trials, it takes a few years. They might cost about $100 million. They're incredibly expensive, they're slow, they're tedious, and 2 big ones are coming to fruition this year. So let's hope that summer and fall of this year we see some promising news. But the point is that look at all these other trials going on. It's a very active area. Okay, so I'm going to wrap up and as I say, turn it over to the proper docs. I just wanted to say that what they're going to cover in some more detail are these things here, the tools, some of the drugs, coping with diseases, disease progression. So if I've whet your appetite but you're frustrated that I didn't tell you much, they're going to tell you more. And finally, I'm going to end by saying it's a sort of high-tech business often. We're developing new instruments. It's very costly. We do some of this old-fashioned sort of chemistry, test tube lab stuff, but we also do a lot of people stuff. We bring groups together. We do a lot of collaborative research. These days, if you're not doing research with lots of investigators around Boston, around the country, around the world, you're probably not doing it right when it comes to Alzheimer's disease. So we're doing an awful lot of that. There's a lot of things here I haven't mentioned, you know, gene chips and animal models and fancy microscopes. It costs an absolute fortune, and at the moment, we are not shy of ideas of what to do to try and beat Alzheimer's disease, but we are absolutely strapped for cash. And so next time you bump into your Congress person or your senator, put them in the corner and tell them about this, and don't let them go till they tell you they're going to spend more money on it, because you saw that tidal wave before. We're at the start of a curve where we're going to have 3 times as many people with Alzheimer's disease over the next 30, 40 years unless we do something about it. And we need to do something about it. With that, I'll turn it over to Risa. So I mentioned that Risa, amongst other things, you heard her bio read out at the start, but Risa, amongst other things, is an absolute expert in clinical trials. She's one of the world leaders in that. She sort of blushes when I say that, but it is absolutely true. And she's also one of the world's expert in imaging, and so she's going to tell you a bit about both of those things. How can we look at Alzheimer's disease before symptoms appear, and what can we do about it? Thanks, Risa.
I'm going to steal the pointer back. All right. I'm vertically challenged, so I've got to pull this down a bit here. All right, great. Well, thank you, and it's really thrilling to see so many people out tonight to learn about Alzheimer's disease, because I think the real way we're going to beat this, so we absolutely need more funding, but we also need more knowledge. So the more people who are interested in this and learn about it, that's the way we're going to really beat it. So I'm going to start where Adrian left off, which is talking about this tsunami or epidemic of Alzheimer's disease. I have some similar numbers, but I want to especially point out this one, which is he showed you that inflection point in the curve. Well, that's because as of January 1st in this year, 10,000 baby boomers turn 65 every day. And because Alzheimer's disease is largely age-linked, and because all of the people who are speaking in all the other mini-med, the cancer docs and the cardiologists are doing such a good job at keeping people alive longer, unfortunately, we really are creating an epidemic of Alzheimer's disease. Now, most people, when they think about Alzheimer's disease, they think about this stage of the illness, dementia. And you're going to hear a lot about this from my colleague, Dr. Ruth Kandel. Unfortunately, at this stage of Alzheimer's disease, where people have really lost memory, they've lost cognitive function in other domains, such as language and visual spatial function, and they lose function, the ability to dress themselves and feed themselves at the very end. And unfortunately, at that stage of the disease, it's not hard to tell what's going on. But we believe that at that stage of the disease, the process in the brain has already been going on for more than a decade and likely more than 2 decades, as you heard from Adrian. So a lot of the research over the past few years has been at an earlier stage called mild cognitive impairment, or MCI. So how many people have ever heard of this idea of mild cognitive impairment? A few, but not too many. So now, sometimes if you go to your doctor and you're complaining about a little bit of memory trouble, people are increasingly using this diagnosis. And it's a little bit of a wastebasket diagnosis in that we don't really necessarily know what is causing the mild cognitive impairment. About 15% per year of older individuals who have mild cognitive impairment do in fact progress to Alzheimer's disease dementia. And after about 5 years, it's about 60% or so that have progressed to Alzheimer's disease dementia. The other 40% or so probably have something else causing their mild cognitive impairment, maybe strokes, other kinds of disease processes that can cause memory problems but are not Alzheimer's disease. Now, I'm going to show you some work that we've done at the stage of mild cognitive impairment and others, and I think this is a tremendous advance that we're moving 3 to 5 years before the stage of dementia. But ultimately, if we really want to fix this disease, I think we're going to have to go back even further to this stage that's now recognized as preclinical Alzheimer's disease. And these are individuals in whom we have evidence, and I'll show you this in a moment, that the disease process is beginning in the brain, but they don't have any symptoms at all. Now you can say, look at this as a glass half full or a glass half empty. So it may be horrifying to think that Alzheimer's disease is beginning in your brain and you don't even know it yet. But what if we could treat it at that stage before people ever got to have any symptoms? And this is our goal. Now I'm going to show you this slide that Adrian showed you, and actually this is a patient that Dennis Selko saw, and I saw this patient with him when I was a neurology resident, and unfortunately this patient progressed, went on to die. He was a very high-functioning mid-60-year-old, I think a stockbroker, unfortunately, and I saw him at the stage that he already had dementia, and again, as Adrian already showed you, this are amyloid plaques, and these are neurofibrillary tangles. And at this time, which was about 15 or 18 years ago, the only way we had of knowing whether somebody had Alzheimer's disease really was after they passed away, and we got to look at their brains under a microscope and could see this. What's really exciting over the past few years is that we now have tools that allow us to peer into the living brain and measure some of these proteins, in particular amyloid when it's in this plaque form. So this is not a PowerPoint malfunction, but a metaphorical black box. And this is what I'm going to be talking about today, is how do we understand how you get from the pathophysiology or these changes in the brain that we recognize begin many years before people get symptoms, and how do we get through this black box and then get to the syndrome that we all recognize and is so devastating to patients and their families. So how can we detect evidence of Alzheimer's disease in the living brain? Well, there have been tremendous advances, as I mentioned, on this, and the biggest one, I think, is the ability to look at amyloid plaques in the living brain. And this is work that was actually pioneered first at University of Pittsburgh, and now we've been working very hard here over the past 5 to 6 years at Harvard to work with these techniques. Now, those amyloid plaques that you saw in Adrian's slide and my slide, you can label those with a dye. And when you label that with a dye, you can now see it using a PET scan. So here is a normal older brain which doesn't have much of this yellow and pink, and that's because most normal older people who have a good memory don't have evidence of amyloid in their brain. This is a patient who has actually relatively mild Alzheimer's disease, so was recently diagnosed with that. And you can see here in yellow and pink, these are where the dye that goes with that PET scan are attaching to that amyloid plaque. And you can see that even at the early stages of dementia, there's already lots and lots of plaque around the brain, and in particular in these parts of the brain that we know are very important for attention and thinking and memory that are so affected in Alzheimer's disease. Well, here are a little bit more data on that. Again, here's a different normal person who doesn't have much amyloid. Here's a different Alzheimer's patient who's a little more advanced, and you can see they've got amyloid plaque really throughout the neurons in their brain or surrounding the neurons. And again, if you do this making numeric summaries of how much amyloid is in the brain, you can see that almost all Alzheimer's patients at the stage of dementia already have very high levels of amyloid. If you look at this stage of mild cognitive impairment, again, it's about half and half, or 60% or 40%. So these individuals who have mild cognitive impairment already have evidence that they've got almost as much amyloid as we see in Alzheimer's disease. But these are the 40% of mild cognitive impairment who have some other reason for their memory problems. They probably don't have Alzheimer's disease. And the research suggests so far that these individuals who don't have amyloid but have memory problems are not likely to progress to Alzheimer's disease over the next 5 years. But the most striking finding in our group and many other groups who've been looking at this is if you look at the normal older individuals, and here's 86 older individuals in our Harvard Aging Brain Study, about a third of them have evidence of amyloid in their brain. And in fact, they have amyloid at the same levels we see in Alzheimer's disease. And here's an example of one of these individuals who is a normal control, absolutely bright, doing very, very well in our tests, but they have evidence of amyloid in their brain in the same pattern that we see in Alzheimer's disease. So what does this mean? Well, here's one idea. If you look at the prevalence of Alzheimer's disease dementia, so again, if you look above age 65, it's about 10% or actually as high as 11 or 12% of individuals have Alzheimer's disease dementia, clinically evident Alzheimer's disease, and this goes up exponentially. So once, unfortunately, you're over the age of 85 or 90, it's 35 up to 50% of people over the age of 90 are thought to have Alzheimer's dementia. Now let's look at autopsy. If you looked at individuals who lived to be age 60 to 90 and were unfortunate and got hit by a bus but were otherwise clinically normal, you can see that this is the percentage of them who have amyloid plaques. And now if you do amyloid imaging, you can see that that maps exactly onto this. So above the age of 65 or 70 here, again, it's about 30% of normal older people already have amyloid. So one possibility is that we're looking at the preclinical stages of Alzheimer's disease. And right now, the estimates are that it takes about 15 years from the time that plaques first appear in your brain to the time where there's clearly recognizable symptoms at the stage of dementia. Now, this may sound crazy and heretical, but most of you understand heart disease, and you understand that lots of us build up cholesterol plaques in our arteries and our blood vessels going to the heart. And lots of people who get cholesterol plaques never have a heart attack, but a lot of people who have heart attacks, if you look closely at their heart, they have evidence that those cholesterol plaques have been building up for many years before they actually get to the point of having a heart attack or a stroke. And although that's a simplistic analogy, perhaps it's the same in Alzheimer's disease. There's a lot we don't know. We have to go back to basic research and translational research. For instance, we now know that there's good cholesterol and there's bad cholesterol. And I bet one day we're going to know there's good amyloid and bad amyloid, but we don't fully understand that yet. So a lot of our research has been trying to understand, well, if you're walking around with a head full of amyloid, can we see other subtle changes in your brain that look like maybe the neurons, the nerve cells, are being bothered, and especially those synapses that you heard about from Adrian? Could we look at evidence of whether those synapses are having early changes in function even before people get any symptoms? And in fact, we can see now using brain imaging changes that look very similar to what you can see under the microscope. So you can see evidence that these dendrites and axons are changing and the connectivity is changing, and we can see evidence of brain shrinkage and changes in function, again, years before people develop symptoms. Now I'm going to show you one example of what we do with individuals who are kind enough to lend us their brains just for a day, not in a bucket, fortunately, at this point. And I got very interested in how we could study memory, and especially develop a memory stress test. So when you go to the doctor, you can get an EKG where they put on the leads. But if your doctor wants to know if your heart is really at risk of having a heart attack before you have one, they put you on a stress test. So we thought we'd develop a memory stress test to be able to look at the brain. And it turns out that learning the names of new people we meet, like you're going to meet outside today after this lecture or the last party you went to, is truly a memory stress test. And in fact, I was so struck by how many people would come into my office and say, I can't remember people's names anymore, Doc, I'm sure I have Alzheimer's disease. I reassured them I could never remember people's names, and that doesn't necessarily mean they're getting Alzheimer's disease, but it really does require that the brain work very well together to form the association between a face and a name. And there are new techniques of imaging using something called functional MRI, where we can actually look at how the brain forms these new connections. So during the time that people are meeting folks at a party, or in our case, we're just showing them pictures of faces and names, we can see what's happening in the brain when they successfully form those synapses and make a new memory, and when they fail. So I'm going to show you some examples. We show lots of faces and names that people have seen. They see them only for a couple of seconds each, and we do warn them that we're going to test them afterwards for which name goes with which face. And these are all unfamiliar names and faces. Now, this is a very difficult task, like this guy here who can't remember his own name. But in fact, it's amazing that when you see one of those pictures for just 2 seconds, you can make this. So who is this? Okay, excellent job. And in fact, I can predict, if you were in an MRI scanner, whether you're going to remember it or not 30 minutes later by what happened in your brain at the time you were learning it. So here's my brain. Here's this part of the brain that's very, very important for forming memories called the hippocampus. And if at the time you saw Lauren, you later, 30 minutes later said, that's Lauren, I'm positive about it, this is actually what your hippocampus did. So it turned on. If you said, I think that's Lauren, but I'm not so sure, you'd see in pink, it goes up a little bit, but it didn't really fire in that way. And if you're like me and said, I don't know, could be Rose, could be Adrian, I have no idea, unfortunately, then they turn you off. Let's see if this will come back on. There we go. Okay, I think I just got overexcited. All right. That's there. So again, this was our first experiment, so we did a lot of these in young subjects, and this was our first chance to say, well, maybe we can look at the brain in early Alzheimer's disease and see when it's working well and see when it first begins to fail. So this is some work at the stage of mild cognitive impairment, and we looked at people who had late-stage mild cognitive impairment, those who went on to develop Alzheimer's disease dementia within 1 to 2 years, and then we looked at people a little bit earlier who were probably 4 to 5 years away from the diagnosis. And again, here's this part of the brain that's so important, the hippocampus, and you can see at the stage of early MCI, it's still working. In fact, it might even be in kind of hyperdrive, so we see increased activation. But by the stage of late mild cognitive impairment, getting close to the Alzheimer's disease dementia, already the hippocampus is failing. It's already not working. And this was evidence to me that if we really want to save people's memories, save them from developing Alzheimer's dementia, we're going to have to go even earlier than mild cognitive impairment with some of these drugs. So this is some work we've been working on at our group here at Brigham and Women's and Mass General, looking at that face name test, and we can see that individuals who are clinically normal but have evidence of amyloid in their brain or a positive PIB scan in this case, they're already performing less well on that face name memory test, even though they're still in the range of normal. And importantly, we can look at whether education or other factors affect this. And individuals who have a lower education or a lower IQ, unfortunately, they're more likely to show evidence of subtle memory impairment in the setting of having amyloid. So that should make you feel all very good, because here you are educating yourselves, using your brain, and protecting it against amyloid right now, we hope. All right, so I'm going to spend my last 5 minutes here in this question, which is, as we've proposed these criteria to move earlier and understand preclinical phases of Alzheimer's disease, there's been a lot of pushback, which is, why would you possibly want to diagnose Alzheimer's disease even a decade earlier if there's nothing you can do about it? But I'm convinced that this is the way we're going to do something about it. If you think about heart disease, diabetes, cancer, stroke, all of these diseases, we've made progress because we treat people before they have symptoms. In fact, when I try to think about drugs that really work after the stage of symptoms, the only one I can think of is Viagra. I think it's really the case that all the huge strides we've made in medicine is by going earlier and thinking about preventative treatment. And in particular, this is important as this is me, because I have a window in my door, and every time I go out and lock myself out and leave an Alzheimer's patient in there, I realize that, unfortunately, our generation is coming. So, again, I don't need to tell you, unfortunately, about the failures we've had in Alzheimer's disease. You can't pick up the newspaper or look on a TV screen without seeing evidence, unfortunately, that many of the large-scale trials we've done in the past have failed. I just updated this last week, or 2 weeks ago, because now it's 10 large-scale phase 3 trials have failed at Alzheimer's disease at the stage of dementia. But at the stage of dementia, we know that already 50 to 70% of those nerve cells in key areas of the brain, especially in the hippocampus, are lost. And I think it's unlikely until we get nerve growth cell or stem cells working that we're going to be able to grow those neurons back. So I think we're going to have to go earlier if we're going after other types of therapy. And importantly, even if we couldn't cure the disease, but just slow it down and delay dementia by just 5 years, we'd cut that cost that you heard about from Adrian of $603 billion in half. So this is part of the reason. This is structural imaging, a different way of using magnetic resonance imaging. And here's a patient with mild Alzheimer's disease. And even though their memory is still pretty good, you can already see that they've lost cells here in the hippocampus. They've got shrinkage all around the brain. And again, treating someone at this stage, I think, is unlikely to grow those nerve cells or those nerve connections back. What we have available right now is what I'll call symptomatic therapy. I use it. I think that's valuable, that we do have some drugs that treat the symptoms, but they're not really changing the course or the slope of decline. And what we're all working on is trying to develop what's called a disease-modifying therapy, which will flatten out this curve and hopefully keep people at the normal or the very, very mildly impaired stage. Now, as you heard from Adrian, there are a number of trials that are ongoing, and all of us have our fingers and toes and everything crossed that we will see a good signal from one of these studies. And there is evidence already published suggesting that we're at least hitting our targets now. This is one of those antibodies. It's an antibody, bapineuzumab, against the amyloid protein that we showed you. And this is some data that was published last year in a very small trial where they had amyloid scans pre- and post-treatment. And here you can see, at least in some individuals, that the amount of amyloid they had at the beginning was significantly decreased over the year and a half of treatment. And this is just a plot of showing you. So here's the placebo group who keeps accumulating more amyloid over time, and here's the treatment group. Unfortunately, though, even though this was a significant difference, we didn't really see any clinical benefit at the stage of dementia. And this also convinced me. Now we've got biologically active molecules, but maybe we're trying them too late in the disease. This is an urgency. In fact, I want to put emergency at the top of this slide because we really need to do something about this. Now, this idea that we could treat Alzheimer's disease 10 years earlier, it's going to take us 10 years to figure out whether we're right or wrong by starting these trials now. And very importantly, just like we do with cholesterol or blood pressure or the sugar test you get in your blood, we need to understand what this means on an individual level. If you have amyloid and you're 77, what does that mean? Will you actually live to be 99 and never develop dementia, or can we give you an individualized risk? I greatly hope, as I mentioned, that one of these studies that we're already doing will give us a signal of efficacy. But in the meantime, I think we need to start these earlier studies. And there are a number of studies that are already beginning, both in genetically at-risk populations for Alzheimer's disease and in older individuals who have evidence of amyloid. And this one I'm very proud to be the project leader of. This is a trial that we've just put in a large grant for 1,000 people who will have amyloid in their brain that we can detect on scans. And we will treat them with, I hope, a biologically active antibody and say, can we prevent the emergence of symptoms before they begin? So who was this? Okay, good. So none of you need to be in that trial at all. I will, however, say that if you are interested or you have loved ones who are interested in trials, we run a number of studies over at the Center for Alzheimer Research and Treatment. And even if you're not interested in being in our studies, there are many studies going around all over Boston. There are studies for normal older individuals, for people who have a little memory trouble, and for people who already have dementia. So I'm hoping that some of you here will be interested in learning more about it. At the Center for Alzheimer Research and Treatment, we do clinical trials in patients who have some dementia or impairment, and the Harvard Aging Brain Study is where we're following older individuals who are normal to try to see if we can keep their brains as healthy as possible. And I'll end there and turn it over to Dr. Kandel. Thank you so much for your attention.
Thanks so much, Risa. That was terrific. So our third speaker, Dr. Ruth Kandel, is also has an academic appointment here at the med school. She spends most of her life as a geriatrician and is director of an Alzheimer's and aging clinic at Hebrew Life. So she's going to take us on the third part of the story. Thank you so much.
So I'm going to be focusing on the clinical aspects of Alzheimer's disease. And what I'd like to do is start off first with what normal aging is. And as was said before, significant memory loss is not a part of normal aging. When we age, we may complain of having memory loss, but that does not mean we have dementia. The most frequent complaint we hear, and we hear this time and again, and I have it myself, is difficulty remembering names, and that is entirely normal. Also, difficulty with tasks requiring divided attention, and these are often more complicated tasks. People have that sometimes as they get older, and most importantly, there's a wide variability in the rate and amount of decline, if any. So now dementia. Dementia is a disease, not a syndrome or a collection of symptoms. It's characterized by progressive cognitive impairment. The symptoms have to be severe enough to really impact on your everyday functioning. And Alzheimer's disease is the most common cause for dementia. Now, I'm going to start with a case. It's a case of Auguste. She's a 51-year-old female who comes in to see her doctor. She's actually brought in by her husband because she has memory impairment. She has aphasia or problems with her language. She has paranoid ideation. She actually thinks her husband is having a relationship with the person next door. And she has difficulty cooking meals and neglecting her housework, and she used to be excellent at this. So you know something has changed. She has cognitive impairment enough to impact on her everyday life, and she has some behavioral problems. So she goes to her doctor, who happens to be Dr. Alzheimer. Auguste presented to him in 1901, and this is actually the first documented case of Alzheimer's disease. Dr. Alzheimer took a great interest in her, and in 1906, when she died, she was admitted to a mental institution in Germany. She died in 1906 from septicemia, or an infection due to bed sores, and he requested her brain for autopsy because at that time he was both a clinician and a pathologist. And he saw some interesting things in the brain. He actually presented it. He called it a peculiar disease process of the cerebral cortex, and what he found were plaques and tangles, and to this day, that is the hallmark of Alzheimer's disease. Now, after this, he didn't become famous. This wasn't considered this incredibly important breakthrough. Alzheimer's disease was considered a very rare type of dementia, because if you remember, Auguste was only 51 years old, and Alzheimer's disease was considered a type of pre-senile dementia. In the 1960s, some researchers got together in England, and what they did is they looked at elderly people who had dementia, and they also looked at their brains and saw indeed there was a correlation between their dementia and cognitive impairment and the amount of plaques they had, and that's what transformed the field. And after that, we've really learned so much more about dementia because now we realize Alzheimer's disease is indeed the leading cause of dementia both in young and old people. So in terms of the statistics, about at least 5.4 million people in the United States have Alzheimer's disease, and by the year 2050, it could be up to between 11 to 16 million people, unless we either can delay the onset of the disease or really prevent it. It's also the 6th leading cause of death in the United States. So how does it present? The early stages of the disease, and you heard about the hippocampus, and that's an area of the brain that really helps us learn and retain new information. And the neuropathologic changes in Alzheimer's disease first impact on the hippocampus and the surrounding area. So the very first symptom you see is this problem of learning new information. So in the course of your day, you might read something, want to remember it, you'll read it perhaps a few times, then you'll go about your life doing other things. And it's at that time then, so to speak, your hippocampus kicks in, encodes that information, and helps you retain it. In Alzheimer's disease, that doesn't happen. So people really can't remember things, and they have a rapid rate of forgetting. That's often why you hear people say, you know, I can't remember what happened 2 days ago, but I remember what happened 20 years ago. The next is language, and early on it's very subtle. They just can't find the right words to use. The next involves executive dysfunction. Executive function is dependent on this part, the frontal part of our brain, the frontal lobes. And in AD, there's just something off. People they have some impaired reasoning, their problem solving is a little off, their judgment is off. It could be subtle. Visual spatial skills. Sometimes the presenting symptom can be problems with visual spatial skills where people are getting lost driving in familiar locations. Auguste got lost in her own home. Functional decline, and at this point it's for more complex functions such as managing your finances, writing out checks. You could have a mood disorder such as depression, and sometimes that could be the first symptom of the disease, but it's sometimes that you need to tease out what exactly is depression and what is apathy, because you can present with AD with just a sort of apathetic, distant feeling that is not a mood disorder. And then the onset and progression of symptoms are gradual. This is something that doesn't change overnight. So that's early stage. Now these plaques and tangles, unfortunately, continue to progress, so the cognitive impairment progresses as well. So now in terms of the memory loss, you have more remote and overlearned information. You can't remember what happened 20 years ago. Aphasia, when you talk to somebody, you can tell just by listening to how they're talking that there's a language problem. Sometimes they have impaired comprehension. They can have what's called semantic paraphasias. An example of this is with Auguste. She was eating at lunch cauliflower, and they asked her what she was eating, and she said she was eating spinach. So she knew it was a vegetable, she couldn't get the right word. You could have circumlocutions where you talk around a word. I had a patient who came in with his niece, and then he didn't know the word niece, and he kept saying, oh, you know that nice person, I've known her many years, but he just talked around the word he couldn't recall. You have executive dysfunction. At this point, it's more progressive. You have difficulty with abstract thoughts, problems with mental flexibility, shifting from one task to another, difficulty just planning your day, and the problem solving and judgments are further impaired. And you have something called apraxia and agnosia. So with apraxia, you have an inability to carry out purposeful movements despite intact motor and sensory skills. So people have a difficult time figuring out how to use objects. Somebody might not know how to comb their hair or how to use a toothbrush, or even doing something in a certain sequence. So just pouring a cup of coffee and then milk and then sugar becomes a problem. With agnosia, you can't recognize something, but your vision is entirely normal, and sometimes it just can't recognize what an object is, or they might look at a face and no longer recognize who that person is. And then way down there is functional decline. And at this point, in the middle stages, you usually need some type of assistance. Here it would possibly be with personal care, grooming, hygiene, but it's hard to live independently during the middle stages. Now, I just want to touch upon the behavioral and personality changes. We talk a lot about the cognitive changes in AD, but there are personality and behavioral issues that can be devastating for the patient and the family as well. During the middle stages, you start to see delusions, which are false beliefs. The most common are paranoid ideations, like Auguste had. Another type of delusion, we talked about agnosia, where the person can't recognize the face. This kind of delusion is they actually recognize the face, but they think that person is now an imposter. So they might recognize their husband, but think that person is an imposter and not really their husband. You have hallucinations, seeing things that are just not there, or hearing things that are not there. Wandering, aggression, and again, depression. And then you have late-stage disease, which is a final common pathway. If you see somebody who's at the end stage of dementia, you really can't tell what type of dementia they had originally, because they're all very similar at the end. So at this point, those plaques and tangles are really throughout the brain. You have memory now severely compromised. Speech is limited. People at this point need 24/7 care. Functional impairment, motor system that was spared before now is involved, and you have bradykinesia, you're slowing down, rigidity, behavioral disturbances, which could be very distressing, including aggression. Seizures can be seen at this stage, and difficulty swallowing, leading to aspiration and pneumonia. Now, I'm going to revisit William Ottermolen. He's quite popular here. And I want you to look at him now in terms of the stages we just talked about. So this would be early stage AD, and on the left, again, this is right before diagnosis. This would be middle stage of the disease, and again, you can tell already he's having visual perceptual problems, and you can get some sense of his emotional affect. And again, distressingly, these are some of his last pictures. Now, I did put in about aggravators of dementia. We talk a lot about treating dementia, starting medications, but there are other things that might aggravate somebody's dementia. It won't cure them if you treat them for this, but it might help them function better. One thing to look at is are drugs. There are many drugs, including over-the-counter drugs people take if they have a cold. This is cold season. They're antihistamines. People take Benadryl to go to sleep, and that can cause acute confusion. There are emotional problems such as depression. Treating hypothyroidism, again, won't cure the dementia, but might help and improve their level of functioning. Eyes and ears, I can't tell you how many times in clinic, all we do is we put on headphones or some type of device so that somebody can hear us, and now they can attend to tasks and their memory improves. Nutrition, trauma, infections, somebody could get a pneumonia and overnight seem like they have an acute change in their status, but then you treat the underlying infection and they're back to where they were. And of course, things like alcohol are not good for the brain. Now, we know that there are a number of caregivers out here, so I was asked to just talk briefly about giving some advice about what do you do when you're taking care of somebody you love who has these problems. In terms of memory loss, structure and routine increases a sense of safety. Using cues, such as calendars, lists, and written reminders, include pictures. Sometimes people have a hard time with written words, but they do really well with pictures. Repetition of information helps. Now, I'm just going to backtrack. What can be very distressing for families is when the loved one repeats the same thing again and again and again. And often people ask us what to do. Now, the first thing, of course, is to know that these symptoms are not intentional. The person is not doing this on purpose. If they had a choice, they would never be doing this. So the key thing is just to take your time, repeat what you said, reassure them, and then try to redirect them. It's called the 3 R's. And then connect with the emotional, not the literal content of statements. If somebody's talking about their loved one, they want to see their mother, where is their mother? You don't have to tell them again and again that their mother has died, but just to know that they may be looking for reassurance, to hug them, and connect to the emotional content. Learning the language of Alzheimer's, keep your comments simple, ask questions that require yes or no answers, answer questions with simple words, and be prepared to repeat the answer again and again, and remain calm. Now, changes in personality and behavior. Connect with the emotional content of the behavior. Use distraction, humor, and music are great. Don't take things personally. Again, this has nothing to do with you. This is the disease process. And don't panic. Enlist the help and support of others when necessary. Dementia is a family affair, and nobody can go it alone. This is really the time to really enlist your family, your friends, religious organizations. The Alzheimer's Association is a great resource. Even going to agencies and hiring somebody just to give you some break, because being a caregiver takes a toll. It takes a toll on people medically, on physically, emotionally, and financially, and it's really a time to get other people to help out. Now, it's not always Alzheimer's. Sometimes there are other causes for dementia. As people get older, often there's an overlap. So you might have Alzheimer's and vascular disease, or Alzheimer's and Lewy body disorders. So this is just sort of a little list of other types of dementias you might see. Vascular dementia, which is due to cerebrovascular disease. Dementia with Lewy bodies, that presents a little differently. People have these visual hallucinations well formed. They might have Parkinsonism, and they might have different problems with their attention early on, and less of that memory loss we just talked about. Frontotemporal dementia actually is the second most common type of presenile dementia and might present with a dramatic change in personality. People with Parkinson's disease don't have to get dementia, but over time some do. And then there are a group of others I just listed. And I'm going to conclude with this. I just want to mention a story. This is Oliver Sacks. And this story comes from The Lost Mariner. This comes from the book, The Man Who Mistook His Wife for a Hat. And just an aside, that particular man who mistook his wife for a hat had visual agnosia. But this particular story is a lost mariner, and it's a gentleman who had severe Korsakoff syndrome. So he had severe memory loss from alcohol abuse for many years. And Oliver Sacks befriended him. He wound up going to his nursing home, and he called him a lost soul. And he was saying this to the sisters at the nursing home, and they said, well, you know what, why don't you come and take a look at him? And indeed, he watched him and said that this gentleman who had no memory, when he listened to music, he transformed. When he took up gardening, it transformed him. He went to the chapel. He was a different person. And he realized that he actually was getting some meaning in his life that he never anticipated. And he concluded this part of his story saying perhaps there is a philosophical as well as a clinical lesson that in Korsakoff's or dementia, there remains the undiminished possibility of reintegration by art, by communion, by touching the human spirit, and this can be preserved in what seems at first a hopeless state of neurological devastation.
So I think what we're going to do now is open it up for questions, and Dr. Spilling and Kendall are going to take a seat over there, and we're going to have a go at some of the questions you've given us. So first of all, I'd like to repeat something that Risa said. There's obviously nothing wrong cognitively with this crowd because these questions are so sharp and to the point that you're clearly all fine. Okay. We got a ton of questions, so I've picked out some of the sort of themes that have appeared here. And here's paraphrasing a few different questions. So can you determine the probability that someone is going to get Alzheimer's disease? Does it run in families? Is it inherited? What are the risks?
Do you want me to take that one?
Want to start with that,
Okay.
Lisa?
All right. I'll start with that. So in rare cases, Alzheimer's disease is completely genetically determined. Only about 2% of cases in Alzheimer's disease, people have what's called a deterministic gene. And these are typically, unfortunately, Alzheimer's patients who develop dementia in their 40s and 50s. Again, these are quite rare, but there is a genetic test for that, but it's only recommended for people who have a clear family history of very, very early onset Alzheimer's disease. In the rest of individuals, we know that genetics contribute. So we know that there's a gene called apolipoprotein E, ApoE4, and different, not mutations, but variations in this gene do increase or decrease your risk. So if you're somebody who inherited an E4 allele both from your mother and your father, that increases your risk of Alzheimer's disease about 5-fold over a person who doesn't carry that genetics. But we also know that there are environmental influences. So even people who have genetic risk factors for Alzheimer's disease, some of them somehow live to be 95 and don't develop dementia. So maybe they have protective genes that we haven't discovered yet. Maybe they eat lots of blueberries and spinach and exercise and stay very active. And maybe they come to Harvard lectures like this.
And Risa, just another one, just to push that a little bit further, but if someone has first-degree relatives, if a grandparent, if both grandparents, if all 4 grandparents have Alzheimer's disease, what does that tell us about our risk?
Yeah. So it's age-dependent, but it is the case that each first-degree relative you have over the age of 65 increases your risk by about 8%. So it is the case that if all 4 grandparents had Alzheimer's disease, that does increase your risk somewhat. But again, there are individuals who develop Alzheimer's disease in their 80s who have no family history. So again, it increases it, but I don't think it's deterministic in most cases.
Thank you. Ruth, how do people die from Alzheimer's? Is it more depression and frustration? Do they just give up the will to live?
No, well, it's a good question. They usually die from complications from Alzheimer's disease. So oftentimes, as we talked about at the very end, people stop eating well. They might have a difficult time swallowing. They become really frail and much more vulnerable to infections. So we often see infections such as pneumonia, especially with difficulty swallowing, people aspirate at the end of life. You might have, in addition to infections, other stresses. People who have had, they just don't have Alzheimer's disease. You often have other comorbidities like cardiac disease, and these may as well be exacerbated towards the end of life. So it's complications from the Alzheimer's disease more than really just giving up.
Thank you. Risa, this person asks, my mother recently had her spinal fluid tested for Alzheimer's disease. What exactly were they looking for?
So the question is about spinal fluid, and we know that there's a fluid that coats the brain that's in all those black spaces that I showed you up there. And you can measure both amyloid, the protein amyloid, not the plaques themselves, but the single protein, and tau in spinal fluid. And it's actually a pretty good test, so that it looks very similar to that imaging in terms of the likelihood of having amyloid in your brain. So 90% of Alzheimer's disease dementia patients have low spinal fluid, which means amyloid, which means it's likely collecting in plaques in the brain. And the advantage of spinal fluid is that you can measure tau, and the combination of amyloid and tau together is more predictive. Spinal fluid, requires getting a little bit of a lumbar puncture, where it's a very small needle is used, but some fluid is taken out. That's used primarily in research, but I do use it clinically sometimes when it's really an unusual case, or if it's a young person, or there are features where I'm not sure whether it's Alzheimer's disease. This can really be helpful to confirm the diagnosis.
Thank you. Ruth, this is a great question. Myth or fact? Doing more crossword puzzles can prevent AD.
Yeah, so there is this thought about use it or lose it, and we do really encourage that. And there have been a number of studies. Unfortunately, there's no great clinical trial looking at this, but there have been encouraging studies showing both physical and mental exercise really does help the brain, and it helps to do things even perhaps you're not great at, like picking up a new language or starting to play the piano if you've never played before. But crossword puzzles are fantastic, as well as physical exercises. Just walking every day for 20 minutes several times a week can help.
And what about dietary supplements?
So dietary supplements is such a huge industry out there. It's a little hard to say to do one thing versus the other. And it's also hard to say, is it taking some magic pill that's going to help, or is it really just eating well, eating green leafy vegetables, blueberries, cutting down on saturated fats, having a healthy style, a healthy lifestyle, and a healthy way of eating rather than specific supplements at this point.
Thank you. Risa, there's a lot in the press these days about genetic testing for this, that, or other, and some of these groups are claiming perhaps that there are some genetic tests. You can take a saliva sample and send it off to someone. Can they test for Alzheimer's with these tests?
Okay. So you can do tests for the risk factors for Alzheimer's disease, such as the one I mentioned, apolipoprotein E, whether you have a certain form of that, ApoE2, ApoE3, or ApoE4. The problem is that it's not, at this point, predictive on an individual level. So if you, again, even have 2 copies of E4, it increases your risk 5-fold, but you could live to be 92 and never get the disease. So I don't recommend it for people, partly because I would tell them to do exactly the same thing. I would still tell them to exercise. I'm not as big a fan as crossword puzzles as I am as going out and start to take ballroom dancing lessons. I think that's a good one. And still try to keep mentally, physically, and socially active. This will change when we have a drug. If we have a drug that really can prevent the disease in people who are asymptomatic, then I think we will see genetic testing play a larger role.
Thank you. Here's another question. This one goes to a question about, we've seen this in the cancer community a lot. You know, a drug is being tested and we can cure cancer in mice, but then it doesn't work out in humans. And someone here is asking the same question. If we've got a drug that is reported as being effective in studies of mice, how long will it take before that might come onto the market? So I'm going to have a go at that one because we run a big mouse lab. There are mouse models of Alzheimer's disease. They are genetically developed, so they carry some of the genes that we think may predispose people to Alzheimer's disease. And they do sort of get a disease that looks a bit like Alzheimer's. So we often use them for testing of potential new drugs. But the number of failures between the mice and the human trials is phenomenal. They are not very good models. They're the best we've got. So next time you read a headline that says, you know, super smart people at Harvard cured Alzheimer's in a mouse, don't get too excited, because at best it's going to be about 10 years plus before you might see that come through. And as far as I know, there isn't a big market for mice with Alzheimer's disease. Okay. Which countries have the highest Alzheimer's disease?
Well, sadly, the U.S. is one of them. So countries that have people live longest and people who have midlife high cholesterol and obesity are actually the highest. The lowest, interestingly, used to be in India. And for a while, that was thought partly because of the diet and in parts of Asia. But it also has to do with genetic factors. So Asians actually have lower E4 allele, and so they actually have a lower incidence of Alzheimer's disease. But in fact, if you account for age and so lifespan, in fact, it's pretty similar across most of the developed countries. So it doesn't actually vary that much geographically.
Thank you. Ruth, is there any connection between Alzheimer's and diabetes? They both sort of seem to be going up in terms of incidence.
Yeah, well, again, diabetes does increase your risk of vascular disease, and cerebral vascular disease and Alzheimer's disease often coexist. And if you treat underlying cerebral vascular disease, you need less of those plaques and tangles if somebody has both cerebral vascular changes and AD changes. So treating anything that would help prevent cerebral vascular disease like diabetes is helpful. In addition, there's a really interesting study that came out looking at intranasal insulin. Turns out that the brain, including the hippocampus, has a lot of receptors for insulin, and they're looking at very seriously treating people. This is all totally in the research phase, but they found that giving individuals intranasal insulin actually helped with delayed recall and function. So there might be more of a connection than we're aware of.
Mm-hmm. Thank you. I'm going to read this one in its entirety. Dr. Sperling.
Oh.
It wasn't clear to me whether it's just so beautifully written. I mean, this is the sort of thing that we ask each other in the lab every day. So whoever wrote this, you should be in the lab with us. Dr. Sperling, it wasn't clear to me whether amyloid is a normal substance in the brain that becomes abnormal in quantity or quality with Alzheimer's. What is the normal function of amyloid in the brain? And then he or she says, thank you, you are great. So what does amyloid
whoever
do?
asked that question is great, because it's a really interesting question. So we don't really know what amyloid, or more importantly, probably its parent protein, amyloid precursor protein, is. We know that all cells make it. We know it's actually one of the proteins that turns over the fastest in the brain, if you're actually looking at what's made and then cleared from the brain. So it probably has some key feature, and most people believe it's somehow involved in learning and memory, and perhaps even in the synapses, but we don't really understand. What we do know is that when amyloid starts to accumulate, when instead of being a nice, happy single amyloid, it starts to form these clumps, not only in plaques, but in these small, what people call oligomers, or these small clumps of 2 or 3 or 5 little bits of amyloid. They seem to make nerve cells toxic. So it's probably like everything else in biology. You need a little bit, but too much is too much. And what we need to understand is how do we keep it at just the right level so your nerve cells work well, but you don't accumulate it.
Thank you.
By the way, that's just like cholesterol. We actually need some cholesterol, but we need to get rid of the bad cholesterol. So I think we'll find that out about amyloid, what's good and bad eventually.
I have to say, I've done this a few times with these question cards, and honestly, I could just pick these up and read them all. They're such great questions, but unfortunately, we don't have so much time. So last 3 quick ones. Someone's asking about disease-modifying drugs and how might they work, and are we trying to inhibit the amyloid, or are we trying to mop it up? And so I'll take a quick crack at that. If we think of amyloid, one way to think about it is the bathtub, and we've got the tap turned on, and there's a ton of amyloid flowing in. And so one approach might be to tamp that down and have less of this bad amyloid. But another approach might be to take the plug out of the bathtub and let it drain away quicker. So we can look at both ends of that equation. But there's another approach, and that would be a sort of vaccine approach. Imagine we could come up with some sort of vaccine approach that identified the amyloid and then arranged for your own immune system that's sort of like bailing out the bathtub. So the point is that, you know, different companies and different research groups are trying all of these approaches. And I should add that they're not just focused on amyloid. There's a lot of other approaches as well that we haven't really had time to go into. I'm going to end on two slightly different questions for either or both of you. What is the effectiveness of private funding, such as the Alzheimer's Association, compared to federal funding, such as NIH? How important is private funding?
Oh, that's an excellent question. So I think private funding is critical for two reasons. One, as you probably know, our government's in trouble right now financially, and so in fact the NIH budget is going down. And although I was thrilled to see that President Obama and so far the Congress has recommended increased funding for Alzheimer's disease, it's still the case that it's actually only about half of the research is funded through the NIH, and the other half comes from private funding, such as the Alzheimer's Association. The other thing is that private funding often will jumpstart ideas. So often if you have an idea where you want to start a study, but you haven't really gotten enough preliminary data to go write an NIH grant, private funding funds those ideas first. And I can say the Harvard Aging Brain Study started with private funding, and that allowed us to get early data, which took us then to get an NIH program project. So without that, these ideas won't grow.
Thank you. So one last point here, and I know you both have something to say with this, so I hope you'll both take 20 seconds to answer this one. But Ruth, maybe you would start. Are there any opportunities for volunteering with AD research? And I know you mentioned before we started that you had this buddy scheme.
There's a lot of opportunities to volunteer for AD research. There is a lot in terms of studies I know that you're involved in. We have some studies for, if there are anyone here who's young, high school students or college students, we've developed a project buddying you up with somebody with Alzheimer's disease, moderate to advanced disease, where you meet somebody for about a week and you really develop a relationship over time. There are a lot of other studies like that that we're involved in terms of clinical work, but then there's also basic research that I know, Dr. Yeah.
We have a study for anyone who's interested, truly, and not just here. So again, we run studies in normal older individuals, mild cognitive impairment, and Alzheimer's disease dementia. But in fact, there are a number of studies going on in Boston, at BU, at many other places. And even if you're not from right around here, you can go on the Alzheimer's Association. There's something called Trial Match. You can put in some information about, I'm a 40-year-old and my grandmother had Alzheimer's disease and I want to do an imaging study, or I want to do a memory study, or I want to be in a drug trial, and they'll find you a study. Right now, the bottleneck is not in the laboratory. We have great drugs that cure Alzheimer's disease in mice. The bottleneck is trying to understand better markers in humans and clinical trials. So every person who volunteers for one of these studies makes a difference, and we'd be thrilled to have your help.
Thank you, Ruth. Thank you, Risa. That question mentioned the Alzheimer's Association, which is just a terrific organization. And if you want to find somewhere where you could help and donate modest funds, they're amazing. So I would just end in that. Now, I know that we're going to have some final comments, but I just wanted to remind, if anyone's interested in seeing a human brain, then we're going to take some time. So we'll probably wait 5 minutes. I know a lot of people need to get off home. But if you want to stick around, then come down and take a seat at the front, and we'll get the human brain out.
Just thank you and good night, and I hope you'll join us for our next program. And thank you to our wonderful speakers. Thank you.